Hello everyone, my name is Ashish Katyal. Today we are continuing our journey regarding Python for biology. This is session 2. In this session, we will start with a short story and after that we will come to our actual part, that is our agenda. So, have you ever heard about a lady that is also known as Typhoid Mary? So, we are starting this little journey of by using this story of learning this Python for biology. So I'm telling you a short story of from 1906 uh, of a lady named Mary Mellon, popularly known as the Typhoid Lady. This particular story is related to a Warren's family, which was wealthy family in New York, and in 1906, 6 people of that family got sick due to typhoid fever, and they are seriously ill due to this bacterial infection. At that time, I am talking about that era in which where antibiotics was not there. So it is important to note down that the era is before the antibiotics, so this typhoid was quite deadly at that time. So that family, Warren family, appointed a sanitary engineer to find out what is the cause of that particular fever. So the engineer name was George Soper, who investigated water supply, plumbing, and other source of contamination, and he found nothing. But eventually, he noticed one thing, that the family's new cook named Mary Mellon, which I have already told you, he checked her history of her employment. So he found that whenever that lady work, there is a case or there are cases of typhoid. So he investigated that thing and he found that lady is a carrier for that typhoid. So she was at that time asymptomatic person that caused typhoid. So you can see during this COVID era, we have seen there are asymptomatic persons who cause COVID, but at that time, that bacterial infection is caused by that lady that is known as Mary Mellon, and she popularly known as that thing. So before going towards this main topic, I am just highlighting this. This is our topic of today, that is Python for biology, and this is session 2. And the lady which I was talking about was Typhoid Mary. So this Typhoid Mary is nothing but Mary Mellon, and she was the lady. And you can see, and you can check this link also. This is a paper that is available at NCBI. You can check that. And due to this particular thing, we can use bioinformatics or Python scripts mainly to find out the DNA sequence, to find whether that particular person is actual infectious, consists of infectious strains of bacteria or maybe virus or not, by using Python script. So it is useful for identifying not only the genes but pathogenic genes also. Also, today's focus of our study is related to find out or writing 5 different Python script for find out the GC content. Why GC content? It is important that the story which I have told you earlier, that at those days we cannot predict GC content, or at that time this technology was not there, but right now we can predict whether that particular DNA sequence consists of the pathogenic DNA sequence, the functional DNA sequence, genes, which is known as genes, consist of any what is the percentage of GC content in that case, because it has been found in studies that the GC content in pathogenic region is quite different from that of the rest of the genome of that particular organism. For example, in case of bacterial species, the GC content ranges from 25 to 72%. But the GC content, for example, in case of Salmonella typhi, which actually cause this typhoid fever, is around 52%. And it is important to note down that the region GC content of that pathogenic region is quite different from the rest of the genome sequence of that particular organism. So the primary focus of our today's study is to write a Python script so that we can find out the GC content of entire organism, that is genome of that entire organism. And along with that, we can use other features like we can find out the complementary sequence of that particular organism. Along with that, we can find out the restriction fragment. For example, you have heard about restriction enzyme like EcoRI, whose site are GAATTC. These kind of thing, these small fragments, there are certain enzymes that cut at specific region. We can find out whether our DNA or genome sequence consists of those regions or not by simple typing a script. We can find out if they are available, how many numbers, at which position they are available. We can find out that. Along with that, if our genome sequence consists of any functional region, we can also find out that. If they are available, what is the length of that? What is the percentage of functional region in our genome? We can also find out that. If they are not functional, that is introns, then we can also find out the length of those intron sequence. All the things we can find out by using this particular Python script. So without any delay, we are starting our session, and you already know in the last session also we have used Anaconda. Under that Anaconda, we have used Jupyter Notebook. So this is the Jupyter Notebook. I have already launched that. So you can see here. So I have already named that under Python 3, and the name of that particular script file is Python for Biology session 2. So without any delay, I am not typing all the things together for saving the time, but I will give you detailed information regarding each script. How can we find out that region, or how can we find out those particular parameters by using Python script. So the first thing which we have to do is related to GC content. So I'm just copying paste that thing, and I will give you detail in that case. So for example, first we have to give the DNA sequence. So DNA sequence, as you all know, in case of for example, I'm just copying things so that I can explain one by one the concept one by one together. So I'm just cutting it and I'm pasting it. So as you can see, the first experiment is related to GC content. You can define a DNA, RNA or protein sequence as a simple string. For defining a string, either you can use a single quote or double quote. So in this case, I am using double quote. So DNA has been defined, and for example, without copy pasting every time, I'm just pasting the full script so that you can easily get the concept, and after that we will explain all the things. So for finding out the length, as in the previous session also I have told you, there is a len command, len, the len command. So for finding out the length of this DNA, I am using this len command, and I am saving my result, that is the numeric result, under this variable that is known as length of DNA. So we can also find out the number of G and C that are available in that particular genome sequence. Why I am doing that? Because the formula says that the percentage of the number of G plus number of C along with divided by total number of length of the DNA, we can calculate the GC content. So this was the formula for calculating the G. We can use a function that is known as count. So for counting the number of G in the DNA, given DNA sequence, I am using dna.count, but we have to count G. So I am highlighting G in a single quote. You can also use double quote also. So also for C, I am using DNA count, but we have to count C. So rather than GC, we can also count A and T. So here I can replace G with A and C with T. We can also find out that. Along with that, GC content formula says that we have to combine both G content, that is this one, along with C content, that is this one, and we have to divide that value with the total length of the DNA. This is the common formula or simple formula. Because we have to find out the percentage, that's why we are multiplying it by 100. So we have to print that thing. So what I am printing, because printing means writing certain strings, I am writing certain strings, it means I have to use double quotes. So in this case, the GC content of the given DNA sequence is, what is that? This is the result, because this result consists of numbers. So I have to convert that numbers into string. That's why I have used str function. So whenever you are writing certain thing, if strings are there, you have to write other things as strings also. But in case of numbers, we have to write numbers. So same thing goes with same. So in this case, I am writing, I'm just clicking shift and enter, and this is giving me the result. That is the GC content of the given DNA sequence is 31.488. You can also change this sequence. For example, you say it is already written sequence, so can I use my sequence also? Or we can use FASTA file also. I will tell you in the subsequent sessions, next session, that is how to upload that FASTA file and perform the same experiment again using that particular thing. You do not have to write every time, you do not have to type every time the DNA sequence. For example, this is my random DNA sequence. For example, I am typing multiple type of GC and ATGC, something like that. And I just have to click on this. This is right now the GC content has been changed to 60%. So this kind of analysis we can do, we can perform by using a simple Python script for finding out the GC content. And GC content is quite helpful in finding out or identifying the pathogenic regions that are available in the gene sequence. So all the things is helpful. The story is quite linked with that. How can we ease the process? Earlier it was not possible, but right now we are doing that. Second thing which I am highlighting is relating to complementary sequence. So complementary sequence, for example, here is spelling mistake it is showing. So I'm just copying paste it, and then I will explain the concept, how it can be possible, how can we use that. So here you can see again the DNA sequence has been defined because it's a string. That's why I use double quote. And after that, I'm just printing this thing. What is my DNA sequence? So I'm printing the given DNA sequences, just comma. Either you can use plus symbol. If you are using plus symbol, then it will not give you a space in between. I'm using comma. If I'm using comma, then it will show you the given DNA sequence with certain space. Then this whole sequence will be printed. And along with that, because the name of that particular string is DNA, that's why I've used DNA. And again, comma, I have to give space, and I'm using /n. /n means new line. The next thing which I want to print, that has certain kind of space in between that. So I cannot write /n directly because it will give me error. So because everything is in string, so I'm using quotes. You will say, you can say that why I'm not highlighting DNA into that is quotes. Because DNA is already is consist of string, so I do not have to again mention that. So the simple formula says that if I have to find a complementary sequence, then I have to simply replace A with T and G with C. If I do this is simple trick which I have shown here. If I do with A with capital T, then again if I am replacing T with A, then it will give me error. So instead of that, I am using a trick, that is I am using a formula, or you can say a function, that is known as replace. First I have replacement shows that I am replacing A with small t. The second then I am printing that thing also, that is first replacement resulted into what? First replacement resulted into changing of capital A with small t. Similarly, I can replace capital T with small a. Why I am not doing the same thing which I have told earlier, that is A with capital T or T with capital A, because it will give me error. So right now I am doing capital T with small a, and same with G with small c and C with small g. All four replacement has been taken place. Right now I am doing that is the fourth replacement. After fourth replacement, it will show me all the letters in small case, but I do not have to use in small case. I want to use in uppercase. So there is a command that is known as .upper command, that will replace all the lower letter into upper letter, and the resulted sequence will be final sequence that consists of all the uppercase. So you can see I am entering it. So you can see the given DNA sequence, this was the given DNA sequence, and in this case this was the sequence that has been printed after replacement of A with small t. You can see all the A has been replaced with small t in this case. Along with that, when we replace second replacement we have done with capital T with small a, then you can see all the small you can say small t has been replaced with small a. The logic was we have to replace A with small t, T with small a, G with small c and C with small g. So here at the end we have to convert with upper. Along with that, the third thing which we have to do is related to restriction fragments. As I earlier told you, there are various enzymes that can cut at various specific end of the genome. For example, this is the whole genome, and there is specific region that can cut. For example, EcoRI is that can is a type 2 restriction enzyme that can cut at GAA-TTC region. So we have to mention this thing. So this is the whole DNA sequence, and we there is a command that is known as find command that can cut at specific region which we have to find. So it is just an analogy that it is finding the region where that particular fragment is available. So +1 because Python starts with 0. So for example, here numbering start with 0, 1, 2, 3. So for finding out the exact fragment length, we have to use +1. So in that case, we have to do. Along with that, in the reverse complement sequence, you can say, for example, here the length is 15. At 5th position, it is available, that GAA-TTC region. But in the reverse region, it is also available because it's a reverse complement. So for doing that, we are just subtracting this first fragment from the entire length of the DNA sequence. So we can get the both length of that restriction fragments enzyme. So this was first is available at 22 position, GAA-TTC, so something like that. When you do the reverse complement, the same fragment is available on the second fragment. So we can do by using a simple formula, that is subtracting from the main length of the DNA sequence, we are subtracting the first fragment length which consists of that site, restriction site. So the fourth one which we have to perform is related to finding out the exonic region, which is also known as functional region, or the coding region that are present in the genome sequence. So it is very simple. Don't look at the formula, this whole script. It is quite big because I split it into smaller fragment. For example, right now we have taken the bigger sequence of DNA, and you can see DNA is a string, and string is donated under double quotes. And first exon, for example, the length of exon we can find out by using NCBI website where the region of exon lies. For example, it is saying it is started at 0th position. This is the 0th position, and it goes towards 75th index point, and we are splicing that. This is known as splicing of the exons. So this first exon has been spliced, this whole region has been saved in exon 1, and we are printing this thing. Along with that, the second fragment lies at 100. So 0 to 75, then there is a gap, and this gap is known as intron. We will cover this in the next script. So then after 100 to 1000, the whole fragment is exonic region. So we are again using splicing thing, that is indexing thing, and this denotes a range operator, which we have earlier used in the session 1. So for finding out the complete length of the coding region, we are just adding both fragments, that is 0 to 75 and 100 to 1000. We are joining them together by using a concatenation command. This is the concatenation thing, and along with that we are finding out the length of that concatenated fragment. So this is the length of total fragmented region, and you can say here, and after that we can find out the total length of the DNA, because we have to find out the percentage of that coding region. So smaller fragments 0 to 75 and 100 to 1000. So we are joining them together, and what is the ratio of this particular fragment towards this whole DNA sequence? So we can find out the total percentage, because total percentage we are calculating, that's why we are multiplying it by 100, and when we click on this particular script we will get the result. So first exon is this one, 75. Second exon is this one, because you can say, sir, we have written 100 to 1000, but this length is not 1000. The remaining sequence, for example, if I can type, for example, 5000 also, or maybe 10,000 also, it will take the rest of the sequence, not till 1000, because 1000 base pairs are not there in this particular case. But if we use the FASTA file that consists of more than maybe 1 million or maybe 5 million base pairs of that, in that case it will work. So in that case I have just used 1000, or you can simply use remove that thing or keep it blank so that it can goes towards the end. So right now for this example I have taken 1000, so it will work in this way. And after that, the total length of the coding region, both the fragments 0 to 75 and the other one, the smaller fragment, it is 98. So the percentage of coding region is 79% in that case. So the rest, you can say that sir, what is the intronic region? So in that case, the rest region, that is 75 to 100. So in case of Python, you already know that it always take the first point, but it goes towards the end, but it not included that particular index value. So in this case, for example, exonic region, it is starting from 0, it goes towards 75, but it only takes the 74th index value. You know, in this case also it starts from 100, it goes towards the end, but it takes the one value less. In that case, I will tell you in more detail in the intronic region. You can see in this case the last experiment or the last script in this case. So here I am putting, so this is the first sequence, this which we have taken, the same thing exonic region, the same thing the other exonic region, and you can see 0 to 75, 100 to 1000. In this case I am taking 75 to 100. So here in the exonic region, 75 is not, it went towards 75, but it is not taking the base pair that is present at 75 position. This is the characteristic of range operator. In this case, right now it is taking 75th position, it goes to 100, but it is not taking the 100 value, it is taking the 99th base pair index value. So in this case, exon, it is starting from 100 till the end of the sequence, same it is there. And right now we are using lower command so that we can distinguish our intronic region from the exonic region. So right now I'm clicking it, and you can see this was the first, same as that of the previous command, and this is the second exon, and this is the intronic region, because it has been changed towards this lower thing, this lower command is used. So all the cases has been changed, all the characters has been changed to lower letters. So in this case the entire sequence is this, and this is the, you can say, the intronic region which is available in our given gene sequence. So these are the 5 different activities which I have told you. that is related to GC content, complementary DNA sequence, restriction fragment length polymorphism, or you can say restriction fragment sites that are available in the given genomic sequence. And we have also sliced out the exonic region. Here in our example, we have done two different exon 1 and exon 2. Along with that, we have calculated the percentage of those coding region, and at last we have not intronic region. We have not only cut the intronic region, but we have changed their cases also. So these were the 5 different things which we have done in our session 2. Thank you so much.